Hi there. My name is Konstantinos Linos, and I'm a practicing pathologist in Dartmouth University in the United States. I'd like to thank the committee for inviting me to give you a couple of talks regarding breast cancer biology and a few things on cord needle biopsies and the chances of upgrade that we're going to talk in the next lecture. So first of all, it's breast cancer biology to give you a kind of basic ideas. How do we define cancer? How do we define breast cancer, the subtypes, the molecular subtypes, etc. I'm not going to go into very deep details, but I'm trying to give you a kind of a basic understanding of where we stand right now. Just book royalties, nothing extraordinary to report. In terms of incidence and epidemiology of breast cancer, this is the new cancer cases in the United States. And you see here that breast is about 270,000 cases a year, and it's the, about 50% of the overall new cancer cases, where in terms of mortality, cancer deaths in 2018, in the United States, the breast was about 40,000 cases, with the highest number was in lung carcinoma, mainly adenocarcinoma, were about 150,000 cases. In terms of estimated deaths, again, it was about 6.7% of 40,000 cases, and the percent of surviving, the 5-year survival rate, with the statistical data that we have between 2008 and 2014, is about 90%. The majority of breast cancers that are being detected today, about 2/3 of the cases are localized, which means they're confined to the primary site. 1/3 of them have regional metastasis, meaning they're spread to the lymph nodes. And about 60% patients present with metastasis, and about 2% are unstaged. So in terms of survival, makes sense that the patients who present with a localized disease, about 98.7%, they have survived, where a regional metastasis, the survival rate drops about 85%, and of course, with distant metastasis, about 1/4 of cases, 1/4 of patients survive. And this is the statistical data I showed you before. The female breast cancers represent about 15% of all cancers in the United States. We're about 260,000 cases in 2018, and about 40,000 cases deaths from breast cancer. The older the women get, the higher likelihood to be detected with breast cancer. The median age of diagnosis is 62, and the most frequently diagnosed, it's most frequently diagnosed among women aged between 55 and 64. Or of course, you can see breast cancers in older women, and of course in younger women. In terms of mortality, the median age at death is a 68-year-old. And the percentage of female breast cancers is highest among women between 65 and 74. So these are the 5-year relative survival data that we have, and you see here was about 75% in 1975, but then introduction of mammography, etc. We now, in 2008, they're about 90%. Now, in terms of breast cancer, there's a lot of heterogeneity. There's a lot of different subtypes, but the majority of breast cancers are adenocarcinomas. And if you remember something in this talk, is this kind of categorization. And based on the expression of estrogen receptor and HER2, basically breast cancer, as it stands right now, it can be divided in 3 major biologic groups. So the first group is estrogen receptor positive, HER2 negative. That's the first group, and it comprises approximately 50 to 65% of tumors. The second big category is the ER positive or negative, HER2 positive, mainly through amplification, which comprises about 10 to 20% of tumors. And the third category, the triple negative or basal-like in molecular categorization, it's the ER negative, HER2 negative breast cancers, which it's the least understood, as we'll show in the further slides, and comprises approximately 10 to 20% of tumors. So this is the big 3 categorization of breast cancer. So as you see here, the ER positive, HER2 negative, the incidence which is the most common, and the incidence increases as quite exponentially as the women get older. HER2 positive is with the purple line, and then triple negative down here. And you see here that the incidence of DCIS kind of increased pretty significantly after the introduction of mammography, and this is the mortality rate of the incidence rate of invasive carcinoma. In terms of risk factors, approximately 90% of breast cancers are sporadic, and approximately 5 to 10% of breast cancers are familial. That means that the patients, they have a germline mutation in a tumor suppressor gene. We have the tumor suppressor genes and then the proto-oncogenes, right? The proto-oncogenes become oncogenic when they get a mutation or a genetic event that amplifies their function, where the tumor suppressor genes, they become oncogenic when they get a mutation or a genetic alteration that suppresses their function, so they don't act as suppressors anymore. So patients with familial breast cancer, which are there are approximately 5 to 10%, they have a germline mutation in tumor suppressor genes, and when they get that, and there are different genes that these patients can be implicated, the lifetime risk to get breast cancer is approximately 90%. So this is the familial, but about 90% are sporadic. So approximately, so this is 10%. So approximately 15 to 20% of women with breast cancer will have a first degree relative, and that is defined as a mother, sister, or daughter. But interestingly, when they do genetic analysis on these patients, they don't find a gene mutation that increase the risk for these patients. So what is happening in these patients is probably they have mutations in low risk susceptibility genes, and that combined with shared environmental factors, they give these patients a higher risk of developing breast cancer. And now there are ongoing studies, pretty significantly deep sequencing studies, that they try to identify candidate genetic variants in these patients that are beyond the BRCA1 and BRCA2, etc. We're going to talk in a little bit. Other risk factors, patients, of course, the race and ethnicity, age of menarche, the age of first live birth, benign breast disease, radiation exposure, etc., etc. These are all risk factors for women to get breast cancer. Now, the etiology and pathogenesis, we have to understand that breast cancer is one disease, but it's not one disease at the same time. What I mean by that is it's a clonal proliferation. You have it starts with a specific clone, but then the heterogeneity becomes quite significant, and that explains why patients that get treatment resistance or behave a little bit different than expected. So they get a few mutations. They have the survival of the fetus. It's Darwin's theory. It's the evolution theory applied in cancer development. And then there is a lot of genetic instability in these clones. And then one other clone will get another mutation, which is different than the clone next door. So that's why we have this cancer heterogeneity, and that's why a treatment may fail or the patients can get resistance. So these genetic acquisitions, this genetic instability, are influenced by environmental factors that they may give a survival benefit to these carcinogenic clones, and that is hormonal exposure, specifically for breast cancer, and of course inherited susceptibility genes. So the familial breast cancer, as I said, is approximately 10 to 12%, and what happened in these patients, they get a germline mutation on one of these major genes, BRCA1, BRCA2. So one gene is not working, but the other one is working. So what happens during their lifetime, they get a second hit on that specific gene. So the two genes, they're not working, so they get breast cancer. So these patients, we have some, they can have multiple affected first-degree relatives. The cancers are early onset. They get multiple cancers depending on what gene is affected, and of course, family members with other specific cancers. So there are clinical indications, there are clinical characteristics that may actually make you suspicious that this patient may have a familial germline mutation, and then they may get referred for further testing. So what genes are being affected in patients with familial breast cancer? Well, the most common one that you know is the patients who have BRCA1 and BRCA2 that are located on chromosome 17 and 13 respectively. And these patients have germline mutations. They have a pretty significant risk of developing breast cancer, up to 90% during the lifetime. And that's why they get prophylactic mastectomy, salpingoophorectomy, etc. But they're not only BRCA1 and BRCA2. These are the most common genes that are being mutated in familial cancers. P53, which is a major tumor suppressor gene, it's another gene that's been implicated in familial breast cancer. And these patients, again, they may have 90% chances to develop breast cancer during the lifetime. And then CHEK2 is another gene who these patients have a less probability, a smaller probability to develop breast cancer, about 10 to 20% during their lifetime. So all these genes are tumor suppressor genes, and the role of tumor suppressor genes in general is actually to repair and maintain the DNA integrity. So when you have an abnormality on these genes, then the DNA cannot be repaired adequately, so then you get a clone that it's kind of carcinogenic and can develop further. So this complete loss of function of these tumor suppressor genes is what we call mutator phenotype. That has been called, what means these clones have an increased propensity to accumulate further genetic damage and then become fully carcinogenic. Mutations of BRCA1 and BRCA2 are responsible about 80 to 90% of single gene familial breast cancer and about 3% of overall cancer. Now, the penetrance is not the same in every patient. The penetrance varies from 30 to 90%, and that depending on the mutation present. The BRCA1 and BRCA2 are very large genes with a lot of different mutations, and some of them are oncogenic, some of them are they're not doing any harm. So depending on the mutation that you have, the penetrance may vary. So patients who have a BRCA1 germline mutation, they actually they're at risk of developing ovarian cancer as well, and that's the rationale when they get prophylactic salpingoophorectomy. Patients with BRCA2 mutation, they have a smaller risk of developing ovarian cancer, but it's been more frequently associated with male breast cancer. Now, as I said, BRCA1 and BRCA2 are very large genes. They're not very easy to sequence them. It takes a lot of effort, and there are hundreds and hundreds of different mutations that are distributed along all these coding regions of the genes. Now, many of these mutations, they're harmless. They don't increase the risk for breast cancer. The frequency of mutation that actually increase the risk for breast cancer is about 1 in 400 persons in the general population. But a lot of them is what we call inconsequential polymorphs. So that means there are mutations that they're harmless. They're in the general population, but they don't increase the risk. So as a result, we just don't do BRCA1 sequencing for every patient with breast cancer. There are specific clinical indication, and because the testing is difficult, it is expensive, and it's generally restricted to individuals who have a strong family history or those belonging to certain ethnic groups. Just to give an example, Ashkenazi Jewish populations, they have about 1 in 40 individuals that carry one of 3 specific mutations. So in that kind of context, then patients will get BRCA1 and BRCA2 mutation analysis. Now, the BRCA1 associated breast carcinomas, they have you kind of correlate a little bit the mutational profile with the morphologic profile that we see under the microscope. So very commonly, they're poorly differentiated. You're not going to see well-differentiated, well-formed glands in BRCA1 associated breast carcinomas. And histologically, they have what we call medullary features. That means a lot of inflammation around the breast cancer. And biologically, they are very similar to the so-called basal-like carcinomas or the triple-negative carcinomas. And we'll talk a little bit further about this. The BRCA2 associated breast carcinomas are also relatively poorly differentiated. And in these cases, they are more oftenly positive for ER than the BRCA1 cancer. Now, the other genes that are implicated in familial breast cancer is P53. So patients who have Li-Fraumeni syndrome, they have a germline mutation in P53. That means that one gene is mutated and it's not working, and they get a second hit on this gene, and they get breast cancer and then mutation on CHEK2. This is about 80%. And of course, other tumor suppressor genes like P10 that's associated with Cowden syndrome, ATM, ST, K11. I'm not going to go into further details on these. Are pretty rare. They are mutated in less than 1% of familial breast cancer. So all these genes, just to give you an idea, they have complex and interrelated roles in maintaining genomic stability. So what happens is we all get mutations, right? Our DNA is being damaged every second and it's getting repaired. So what happens is one of these genes senses that the DNA is not, it's defective. So P53 stops the cell cycle, so it doesn't divide further. There are other genes come and then repair the DNA, and then the cell either dies if the repair is not good, or then goes back to the cell cycle. So there's a very complex and very eloquent machinery there. When we have defective in one of these major genes from these major pathways, then we may get a clone that is carcinogenic and further genomic instability, they get further genetic events. So the interesting thing is we talk about BRCA1 and BRCA2 in breast cancer, but we don't actually know why patients who have a germline BRCA1 or BRCA2, they're more highly associated with breast cancer than other cancers. I mean, BRCA1 and BRCA2 are major genes that are implicated in DNA repair. So if you have a germline mutation in BRCA1 and then you get a second hit, why you don't have a higher possibility to develop other cancers, because the DNA can be damaged not just in breast cells but in other cells in our body. So we don't actually know. What we know is that BRCA1 and BRCA2 are very large complex proteins that they regulate chromatin structure. So it's possible that there are tumor suppressive roles that involves functions that are independent of DNA repair. So they may, which is more specific for breast tissue. So it's possible, it's the reason why these patients have a higher likelihood to develop breast cancer is not associated with the function of the BRCA1 gene or BRCA1 or 2 genes in relation to the DNA repair, but it may be associated with other functions that are more specific to breast tissue. This is to be determined, but it's kind of interesting. Because BRCA1 and BRCA2, as I said, is expressed ubiquitously, which it's expressed in many different parts of our body. So we don't have a very good explanation yet. What I just told you is just a theory. In terms of sporadic breast cancer, which constitutes about 90%, the familial breast cancer, as I said, is 10%. The major risk factors are related to hormonal exposure, and that means gender, of course, women, age and menarche and menopause, reproductive history, breastfeeding, and exogenous estrogens are all related to alternating the risk of developing breast cancer. So how the hormonal exposure may actually increase the risk of getting breast cancer is probably because they increase the number of cells that can potentially give rise to breast cancer. You get more cells, you get more DNA repairs, you get more statistical chances one of them to not to go bad, and you have more statistical chances this to not be repaired and then gets breast cancer. It's all about quantitative statistics, if I can use this oxymoron here. Because once you get proliferation of breast epithelium, it's conducive to accumulation of DNA damage, and more cells you have, the more chances is to develop breast cancer. And once you get this premalignant or malignant cell, that means this genetic alteration that gives the survival, benefit, then this is an unstable cell, and then you get more hits, and become fully malignant, and of course, the surrounding normal stroma cells, they actually may aid in tumor development, and that is kind of a pretty intense research area here. What is the role of the microenvironment and the tumor environment in terms of tumor development? Now, the three major genetic pathways, this is, again, if you remember something of this talk, this will be, and that applies familial and sporadic breast cancer, ER positive, HER2 negative, dominant pathway of breast cancer, 50 to 65% of the cases, HER2 positive cases. How do we get HER2 positivity? Is strongly associated with amplification of HER2, and I'm going to explain you in a couple of slides what that means. So that means you have amplification of the HER2 gene, so you have overexpression of the protein, and then you have the triple negative, ER negative, HER2 negative cancer, which is an independent independent of ER mediated changes. It's a different beast. You don't see HER2 gene amplification for the most part, and this is the least understood breast cancer subtype. Now, the ER-positive, HER2-negative, this is the most common subtype, and in terms of familial breast cancer, you're going to see more commonly individuals have BRCA2 rather than BRCA1. And they have other genetic abnormalities. They have chromosome losses of 1q and 16q, and usually they have activated mutation of this gene, the PIK3CA, which actually is an important component of signaling pathway downstream of the growth factor receptors. So it's below the cellular membrane. It's along the growth factor receptor signaling, and it's kind of an important gene, and that's been alternated in ER2, HER2-negative cancer. The same genetic lesions are found in lesions that are being called flat epithelial atypia and atypical ductal hyperplasia, and the same exactly genetic aberrations are found in these two lesions. And these cancers is called luminal cancer in the molecular classification, and then depending on the proliferation rate, it's called, we subcategorize them in luminal A and luminal B. The HER2 positive cancer, that's the second major category, is associated, as I said, with amplification of the HER2 gene. That's about 20% of cases, and they have a different genetic, a different gene expression pattern than the ER positive, HER2 negative cancers. And in terms of familial, patients with P53 germline mutation, they're most commonly, this subtype is most commonly associated with. The putative precursor lesion for HER2 positive breast cancers is lesions that we term typical apocrine adenosis that kind of have the same genetic background. And then the triple negative cancer, the ER negative, HER2 negative cases, it's a distinct pathway, different from the previous two. It's about 50%, and patients who have BRCA1 mutation, germline BRCA1 mutation, are most commonly going to get triple negative cancer. And this, in terms of mRNA expression, molecular classification, they have a myoepithelial phenotype, or the so-called basal-like pattern of mRNA expression. So this is a diagram of what I presented you so far. You have the normal breast. You have BRCA1, if you know germline, if you have germline mutation, BRCA2 mutation, you have additional genetic alteration, and then you get flat epithelial atypia. Then you get further mutations. This is genetically unstable, prone to get more hits. You get mutation in the PIK3CA, get a typical ductal hyperplasia, you get further genetic events, you get DCIS. This is the luminal pattern. The luminal category, ER positive, HER2 negative, about 50 to 65% of breast cancers. Now, this is the HER2 positive amplification. If you have germline, most likely you're going to have a P53 mutation. You get amplified of this gene. You get a typical apocrine adenosis. You get further genetic events. You get DCIS, and then you get invasive cancer. And this is the HER2 positive cancers. And then the ER negative pathway, it's a triple negative. We don't know exactly what is going on in this kind of subcategory, but you find very commonly P53 mutations, BRCA1 inactivation, and then about 50% of cases are going to have this triple negative or basal-like breast cancer. Now, deep sequencing, what I mean, they did further next-generation sequencing of breast cancers to try to characterize more genes that are associated with breast cancer, and try to reconstruct the genetic events, the sequencing of events that's happening in breast cancer, and they have found other driver mutations. What I mean by driver mutation is, if I get cancer, and they're sequencing my genome, they're going to find a lot of different mutations. But many of these mutations are irrelevant. They are what we call passenger mutations. They are not causing harm. They are not very critical in developing of cancer. So they're noise. And one of the hardest thing is not just to identify the mutation, is to identify the functionality of this mutation in terms of cancer development. If you find 100 mutations, that doesn't mean that if you're going to hit these 100 genes, you solve the problem, because out of these 100 genes, the 90 may be passenger, relevant. So the ones that actually are important in driving carcinogenesis are what we call driver mutations. So these can be proto-oncogenes, and these are some of the genes that are implicated in breast cancer. And these are tumor suppressor genes. Some of the genes are implicated in breast cancer. So the future of cancer in general is actually to transform into kind of a chronic disease like HIV. What I mean by that is with targeted therapy is to target different signaling pathways. They'll get resistant and then target the next pathway, target the next pathway, target the next pathway, and try to develop kind of a chronic disease, although we're pretty in a fetal stage, but that is the rationale of identifying all these different signaling pathways and study the mutation of the cancers. So this is a very important concept. Breast cancer, even the one cancer in one patient is not one cancer. There's a huge subclonal heterogeneity. So different cells have different genetic events. They may have the same initial event, BRCA1, BRCA2, or this or that, but then they go their own way in terms of evolution. So that's why the tumor progress, and that's why many of their therapies they get resistance to therapy, right? Because our therapy is going to target one subclone, but it may not be effective in a different clone of the same cancer. What may surprise you, I mean, that's a lot of work that's happening in the last 5 years or so, that we don't know a lot about the local microenvironment and the interaction of stromal cells and breast cancer. Cancers, of course, occur in the areas of greater mammographic density, and this suggests that the increased amount of fibrous stroma is both a marker of risk and biologically important for tumorigenesis. But it must be very important because the same events that we see in situ carcinoma are in the invasive component as well. Exactly this. So there's something we're missing there. What drives from DCIS to invasion? We don't actually know. That's an area of intense research because there's a lack of understanding there. Because the same genetic events you can see in situ and invasive carcinoma. So this is the final step. That's the most important step, right? The transition of an in situ carcinoma to invasion. It's the most important because that would drive prognosis and change therapy, etc. And it's the least understood. And as I said, the majority of genetic changes that are observed in invasive carcinoma, they're already present in situ. So that is that's a concept that's a lot of things to be learned here. And we'll see what's happening in the next decade or so. So in terms of types of breast carcinoma, about all of them are adenocarcinomas. I mean, we have a lot of different subtypes of carcinomas. And the terms ductal and lobular, they're a little bit archaic, but are still being used because the way they resemble under the microscope, or they resemble ducts or they resemble lobules. But the thing is that most evidence suggests that all breast carcinomas, they actually arise from the same cells in the terminal duct lobular unit. So DCIS is a malignant clonal proliferation that by definition is limited to the ducts and lobules and is surrounded by basement membrane. It hasn't broken the basement membrane. It's not invasive. And the two major architectural subtypes, we call it Comedo DCIS and non-Comedo DCIS. And of course, a nucleograde or grade this and necrosis are actually better predictors of local recurrence and progression to invasion rather than the architectural type. And this is what we call Comedo type DCIS with this kind of necrosis in the middle. This is the DCIS here. And this is cribiform DCIS, have these 9 holes here, pretty uniform. This is a cribiform DCIS. Solid type DCIS, this is how it looks on the microscope, and then this kind of Romer arches, and then some cells floating here in the middle, so-called micropapillary DCIS. This is DCIS kind of filling the lobules, resembling LCIS, and then this is kind of more pink cytoplasm. You see it looks pretty different from the pictures I showed you before. This is kind of a more apocrine type DCIS. Paget's disease is a rare manifestation of breast cancer. It's about, you see about 1 to 4% of cases, and what happens is the cancer, the DCIS, involves the lactiferous lobules and kind of extends into the skin. This is the epidermis. Here, this is the stratum corneum. You have DCIS underneath, and then you have cancer cells colonizing the epidermis. And the most, it looks like eczema clinically, and the most common biopsy is going to be eczema versus Paget's disease. That's the most common question when we get a biopsy. Now, the natural history of DCIS is kind of difficult to determine, and the reason is because I think recently all women with DCIS were treated with mastectomy, so you couldn't follow the natural history of the disease, and mastectomy is largely curative. The patients who have a high-grade or extensive DCIS are believed, of course, to have a higher risk for progression to invasive carcinoma. And the other interesting thing, if you actually see carefully the statistics, you see that the overall death rate of women with DCIS is lower than the women in the population as a whole. It's kind of a contradictory, but that is probably reflects that mammographic screening is a marker for better access to medical care and other socioeconomic factors. It's kind of an interesting statistical observation there. Now, the risk is about 1% per year to develop invasive cancer, and the majority of these invasive cancers in women with DCIS, they will occur in the same quadrant, and they have a similar grade and expression pattern of ER and HER2 with the associated DCIS. Now, death for metastatic breast cancer after diagnosis of DCIS, about 1 to 3%. Of course, you will tell me how is that possible if the cancer is in situ. There are a couple of explanations for this. First is it's sampling. Pathology is an art of sampling. We don't put the whole tissue. I mean, this is going to take 1 year to just do one case. So it's possible these are missed microinvasive carcinomas that were not sampled, or it's carcinoma in the contralateral breast in women with DCIS. But that's what explains that statistical finding. What is LCIS? LCIS is a clonal proliferation of cells within ducts and lobules that they grow in a kind of discohesive fashion, and there is a biological explanation why the cells are discohesive. And because they're losing, we can check this by immunohistochemistry, the adhesion protein, which is called it-cadherin. So there's loss of it-cadherin, so the cells cannot stick to each other very well. That's why they're very discohesive by morphology. Now, the cells of atypical lobular hyperplasia, LCIS, and invasive lobular carcinoma are morphologically identical. And this is how it looks under the microscope. A very uniform proliferation within the ducts. And then if you do it-cadherin, you see here that it's positive in the surrounding cells and myopithelial cells, but it's negative in the actual tumor cells, and that's definitional for LCIS. Now, LCIS is a risk factor for invasive carcinoma, and invasive carcinoma in women with LCIS will develop about 25 to 35% overall 20 to 30 year time, about 1% a year, and it's kind of similar to that observed with untreated DCIS. There are rare variants of LCIS that they look different. They have a high-grade nuclei, have a central necrosis, and these are different. The risk is different, and this should be treated as DCIS rather than as LCIS in terms of management. So this is a nice plot to give you the basic understanding of the subcategorization of breast cancer. ER positive HER2 negative breast cancer, 1. HER2 positive breast cancer, 2. Triple negative breast cancer, 3. And you see here, this is called Circo's plot, and show you the genomic instability of the DNA. These are the lines are chromosomal rearrangements. So basically genomic stability or instability. See here that the ER HER2 negative, that they have a low proliferation index, you only have one line. That's pretty stable. ER expression, and that's why they look pretty low grade. You have nice, well-formed glands, and this is the mRNA expression. So the red is our overexpression of proteins of mRNA. This is down regulation of mRNA, the green. So you see here the patterns are different because they're just different categories. The ER positive HER2 negative high proliferation, luminal B, you see here how way more unstable it is, and it looks uglier under the microscope. And then HER2 positive, and then triple negative. You see here, triple negative is all over the place, a very unstable cancer. That's why it's more aggressive. ER positive HER2 negative, also termed luminal. Again, luminal A and luminal B, they're being defined as low proliferation and high proliferation. The high proliferation is about 10% of the luminal A. The most common type of cancer that is detected by mammography is the ER positive HER2 negative luminal low proliferation rate. This is the most common that is treated with, the patients are treated with resection and then get tamoxifen. And this cancer, actually, they have a very well prognosis, but when they metastasize, they can metastasize after 5-10 years down the line. And even when they get a metastasis, they may actually have a long survival, even with metastatic disease, even when actually chemotherapy doesn't work very well in these cancers, because presumably they have a low proliferation index, and chemo doesn't work with cancers that they don't develop fast enough. And then the high proliferation rate is a different beast and grows faster. The HER2 positive cancer, about 20%, they have more complex rearrangements, as I showed you in the previous slide, and they have a high mutational load. And how do we actually test for HER2 amplification? There are two ways. You start with immunohistochemistry. If this is the HER2 gene on chromosome 17, and the CEP is the centromere of the chromosome. So you can do the immuno, but then the most reliable study is to do FIS. And how does it work? If the gene is not amplified, I have two copies, right? Two copies of the gene. And then I put a prop here and here, so I have two red. And then I put a prop here and here for comparison, so I can see if it's amplified or not. So this is a cancer that is not amplified. I have two genes of HER2, two chromosome 17. I have two greens, two red. But this is an amplified cancer, so I have two chromosome 17, so I have two centromeres. But then I have lots and lots of HER2 genes, so I have lots of red. And it's a very simple thing once you know it. And the triple negative cancer, it's a major molecular subtype, approximately 50% of cases. They share the same genetic similarities, have genetic similarities with a series of ovarian carcinoma, and these kind of tumors, they have overlap molecular subgroups. When I look at the slide, how do I grade? This is the Elson and Ellis modification. I look at the nuclei. They're uniform. They're similar to their surrounding cells. Look mitosis, etc. And then what is the Glanzrow score? And then I came up with a score. So and then I say well differentiated, moderately differentiated, or poorly differentiated. And this well differentiated, you see well formed glands. That's how it looks on the microscope. Where here it looks so different. You don't have glands. The cells have this very prominent nucleoli. It just looks very different than this, and this is a poorly differentiated breast cancer. Now, there are special histologic subtypes of invasive carcinoma that they have distinctive morphologies. They look very different to each other, and they have a relatively unique biologic characteristics. These cancers, like the breast cancer of no special type, they can also be organized into groups based on ER and HER2 expression. And often these other subtypes, they have unique genetic aberrations that could be targeted in the future. And these unique genetic alterations with distinct gene signatures may actually have frequently show an association with clinical behavior and prognosis that sometimes break the rules. So here it looks different, like a Singer ring adenocarcinoma, mucinous type adenocarcinoma, secretory type adenocarcinoma, apocrine. So a lot of different subtypes of breast cancer. The outcome of women with breast cancer, of course, depends on the biologic features of the carcinoma. What I mean by that is the histologic subtype and molecular type, and of course, the extent of disease, how much has the cancer been spread. And these, the prognostic factors, they fall into two groups. Those related to the extent, what is the stage of the tumor burden, and those related to the underlying biology. And the prognostic factors related to the extent, of course, as you know, carcinoma in situ, invasive distant metastasis. The most important prognostic factor for invasive carcinoma in the absence of distant metastasis, of course, the lymph node metastasis, the tumor size, lymphovascular invasion, etc. And in terms of the prognostic factors that are related to tumor biology, what is the molecular subtype? Is it triple negative, HER2 positive, etc.? Is there a kind of other special histologic type? What is the histologic grade, proliferative rate, ER/PR, and HER2 status? And of course, you see here the 10-year survival in terms of stage changed pretty dramatically. Again, the same thing here. As stage 1, 2, 3, the overall survival it's very different in patients that start with the different states. So the key concept of what I presented you today is that breast cancer is the most common non-skin malignancy in women and the second most common cause of cancer deaths. First one is lung carcinoma. The most important risk factors are estrogenic stimulation and age. And all cancers arise by the accumulation of DNA alterations and epigenetic changes. What I mean by epigenetic changes is it's not only your DNA that it may cause, the damage of your DNA may cause cancer. But there are other cancers that your DNA may be fine, you transcribe the protein, but the protein doesn't work because you have epigenetic, what we call epigenetic changes. So mutation profile is not the holy grail of cancer analysis. There is different steps of analysis in terms of cancer research. Tumorigenesis also requires changes in the normal supporting cells, and that is intense research here. The hormonal milieu of breast plays an important role in expanding populations. We have potential precursoral cells altering stromal and proliferation of cancers. DCIS is treated locally. Subsequent invasive carcinomas usually occur at the same time. However, LCIS confers a bilateral risk. So it's a risk factor. Invasive carcinomas have been classified in molecular subtypes based on expression of hormones, ER/PR, and HER2 amplification or not, along with the Ki67 proliferation rate. And the molecular subtypes of breast cancer have an important clinical, biologic, and therapeutic associations. Special histologic types of carcinomas have a distinct pathways of tumorigenesis that they may actually provide additional clues linking biological changes to clinical behavior. And the prognosis of these women depends on the biologic type of the cancer, what is the histologic type, what's the molecular classification, and of course the extent of the cancer, what is the stage of cancer. Effective treatment requires both local and systemic control of disease. And of course, improvements in treatment are being made as new targeted therapies are being developed every month, and response to treatment is better understood. And that's what I have for you. Thank you very much for your attention.